good. All right, so second up today, we have Dr. Edward Lemke, professor at the Johannes Gutenberg University Mainz and an adjunct director at the Institute of Molecular Biology in Germany. Dr. Lemke's academic career began with a diploma in chemistry from the Technical University of Berlin in 2001, followed by a Master's of Science from the University of Oklahoma. He then pursued his doctoral studies at the Max Planck Institute for Biophysical Chemistry, and following his doctorate, he did a 3-year postdoc at the Scripps Research Institute from 2005 to 2008. He then led an independent research group at EMBL in Heidelberg from 2009 to 2020. In 2018, he started his professorship of synthetic biophysics at the Johannes Gutenberg University Mainz and became an adjunct director at the Institute of Molecular Biology. In 2019, he co-founded Viraxa Biotech, a company dedicated to antibody engineering against cancer. The central theme of Professor Lemke's research revolves around the study of what he calls the dark proteome of intrinsically disordered proteins, which lack fixed three-dimensional structures yet play crucial roles in cellular functions and diseases. His laboratory employs synthetic and chemical biology techniques to investigate the dynamics of these proteins by engineering designer organelles for localized encoding of synthetic amino acids into proteins, thereby enabling specific labeling and visualization of IDP conformational changes within living cells. He has won more awards than we have time to list here, but this includes in 2022, he was elected as an EMBO member, and in 2024 as a BPS Society Fellow. Dr. Lemke, thank you for joining us today. It's a great pleasure to have you. Please take it away.
Okay, well, thank you very much for organizing this event for the community and for giving me a chance to present here. I guess I'll just get right started. What you see here is an illustration that one of my more talented co-workers draw. Let me actually change the cursor here. It's an Oreo cookie, interacting with a piece of RNA. And I hope I managed to get there in time to tell you a little bit on why we came up with that illustration. And of course, I have nothing to do with the company Oreo. I'll start with something that we're very familiar with, labeling methods in biology. And I like to remind you that there are these technologies that have the large size, and of course with this community, it's very easy to convince people that there are alternative tools to GFP labeling where you can genetically encode the chemistry and then click something to it. And the smallest modification, I think, is allowed by genetic code expansion thereof. And what you could also plot here instead of size of modification is, I think, to be fair, complexity or activation energy needed to implement the technology into your lab. And I think GFP is really easy, and unnatural amino acids are a little bit more tricky. And I guess it's the goal also of GC community to make this easier. One of the contributions that we did to this field is to genetically encode some of these chemistries that I show here, double and triple bonds. Just to get this very clear, the chemistries themselves were, of course, invented by much smarter people. Among them, Bertozzi got the Nobel Prize for this click chemistry here, and others for the inverse electron demand Diels-Alder reaction. We, together with Carsten Schulz, a former colleague at the EMBL, who's now in Portland, so very close to the GCE facility, genetically encoded these lysine derivatives, and this also called tetrazyme ligation. They really offer the possibility to have reactions quicker than the pipetting speed of many of the students in the lab. Chemistry is quicker than biology in many cases, and we and others have shown that we can have multiple combinations of azide, tetrazymes, and different double and triple bond systems, and could essentially undergo multicolor labeling reactions. And then the question is, how can we actually perform these multicolor labeling reactions inside the living system? So this slide, I usually spend a lot of time explaining, but I guess with this audience, I'll cut it a little shorter. Genetic coding of paralysin tRNA synthetase. I should also say that I use cytoplasmic paralysin system, genetic code expansion, amber suppression. These three terms frequently interchangeably. Just to remind you, we have a stop codon. Of course, we put the tRNA and the synthetase in, and then the synthetase is programmed to only accept our natural amino acids of choice. And when you do this proper the stop codon is suppressed, and you can purify full-length protein, for example, cleanly by pulling on the C-terminus. Yeah. You see this method to work very well in E. coli. You also see it in eukaryotes, but if you go to actually higher resolution methods, you don't see it that often. Also, very physiological studies sometimes shy away from it. And one of the reasons is that we have actually 20% of amber codons in our proteome or in our, RNAome, in our transcriptome. That means that when we suppress a targeted stop codon, we actually suppress, up to 20% of all other codons. And when we put a label in, we start seeing the label, and it starts messing up our signal. In E. coli, this has been beautifully solved. I mean, I think one of the first papers was here by George Church, where they started, to create this field of, genome engineering, which has meanwhile become a huge field with many, many labs making fantastic contributions to it. And here the goal is that if you take all of the stop codons out, the amber codons out, and for example, replace them against an opal codon, then you get a host that is genetically clean. And when you put a stop codon into your protein of choice, only your unnatural amino acid will go there. Yeah? In the beginning, they needed €1 million of primers. Meanwhile, this has come dramatically down, and they can do this at much lower cost. In eukaryotes, talking about low cost, we still can't do this. Lots of chromosomes, much bigger DNA. We don't have a really good way to solve this problem cleanly in eukaryotes. What we have in eukaryotes is that we've now realized that, the process of phase separation actually gives rise to all of these membrane-less organelles. The most famous one maybe the nucleolus, not the nucleus, but these little dots inside. And in those machineries, you basically have extremely high protein, also RNA concentrations, and that probably allows for unique biochemistry and allows those machineries to do specific processing. So ultimately, in a eukaryotic cell, those type of membrane-less organelles, and also, of course, membrane-encapsulated organelles allow you to execute specific reactions that you don't want to have just going wild in the whole cytoplasm or nucleoplasm. And we thought, wouldn't it be nice to have a protein or engineering organelle to follow the same concept, in where you do in the organelle, you do the protein engineering, and you don't mess up with the rest of the host. And we would like to call such an organelle the orthogonally translating organelle, in OTO. Okay. So how would that look for the cytoplasmic system? Let's say I have 2 RNAs now. This now is a canonical one that has a stop codon which shouldn't be suppressed. And then I have a stop codon and an RNA that I want to suppress. I can mark this RNA on the RNA level with, for example, MS2 loops. That would mean that the protein doesn't know it, that this RNA was marked, this RNA with MS2 loops, because they're not translated. And then, the normal genetic code expansion system, the cytoplasmic paralysing system, is, stupid and can't distinguish between these 2 RNAs, and will just suppress both stop codons, and both proteins will be made. That's the one that I wanted. That is not what I wanted. At least this part here was extended to a canonical protein and screwing up the proteome, making an erroneously long protein, and from a fluorescent point, even worse, put a labeling site in here. Yeah. So what we thought we can develop is that we develop a membraneless organelle. And that membraneless organelle needs to have the following features: that the tRNA needs to, our suppressor tRNA needs to come in very close proximity of the stop codon. Yeah. And how do we achieve this? The tRNA is not that easy to engineer, but proteins are easy to engineer. And what we do is we take a protein that binds MS2 loops, for example, the MCP, and we take our synthetase, and to both of them, we basically put a glue, some form of assembler domain that allows them to assemble. In this way, we force our RNA always in local proximity of our synthetase, and our synthetase binds our tRNA. And then you hope that the rest of the translational machinery, another roughly 100 factors plus giant ribosomes can, get access to this machinery. Yeah, that's the beauty here of thinking of a system that's not membrane encapsulated, because if you would put a membrane around it, you can of course get very clear separation. but then how do you get the ribosome in and out in the whole machinery? Yeah. Here we think of a system that is, based on phase separation. So we basically, co-phase separate or co-condensate proteins that are known to phase separate so tightly together to build an organelle where we have the millimolar concentrations. There's no boundary. And we hope that inside the concentrations here, we get also the millimolar concentrations of our suppressor tRNA. Of course, the suppressor tRNA can diffuse outside the organelle, same way we want ribosomes to diffuse inside the organelle. So we don't expect from such a system to be infinitely tight. No, if you want it to be infinitely clean, you need to put a membrane around it. But then again, you also need, again, a transport system. Yeah. How do we test for such a system? You need brilliant co-workers. Gemma and Christopher started this in the lab. We take 2 fluorescent proteins, we put stop codons in there, and you only see the fluorescent if the stop codon gets suppressed. And then only one of our fluorescent protein contains the MS2 loops. So ultimately on FACS plot, where you look for red fluorescent and green fluorescent, you should get something diagonal for cytoplasmic pyrolysis system. And when you have a cell that actually would have a functional orthogonal organelle, only the protein should be made that has the MS2 loops. Yeah? And we tested phase separating proteins. There are many known now. We also tested other means of gluing things together. There's more than phase separation in biology. We also picked, for example, cytoskeletal elements where you can take microtubule motors that can't fall off the microtubule, so they accumulate at the end. And in the end, we also combined both methods of protein concentrations, that means phase separating domains, with those that bind microtubules. Yeah? We tested all of them. This is a busy slide. You don't need to understand all of it. We basically tested all combinations that we could think of highly concentrating proteins in the cell. And then we look at 2 things. One of them is efficiency. That means how efficiently is the amber codon suppressed. And we define 100% as our cytoplasmic pyrolysis system. And you can see here that, we easily get nearly 50%. And then we ask for selectivity. And that is basically on how much we get our mRNA translated that has the MS2 loops in preference to RNA that doesn't have the MS2 loops, for example, the GFP. And you can see that we do get organelles which have significant selectivity enhancement. And one of the beauties is that when you have such an organelle, in such a system, you only need to change the anticodon of the tRNA and the codon in the RNA, and you can even reprogram it to another codon. If you would do this with the George Church method in E. coli, you would need another €1 million of primers to redesign the entire synthetic genome. And I should also say, I also bring some of the data in a few slides, that meanwhile we have achieved, up to 20, 30-fold selectivity. That means every 20 RNA is made that we don't want to suppress a stop codon. Yeah? But the other 19, only the ones are suppressed that we want. And we have meanwhile recovered nearly 100% efficiency. For those that don't like FACS plots, I also show imaging data. So here we have a cellular system and where we do genetic code expansion of 2 proteins at the same time, a nuclear envelope marker and a cytoplasmic protein. They both have the amber codon. They're both made, but if you take the same construct and make it one that expresses an auto, then only the one that has the MS2 loops labeled is made. Here, the nuclear envelope marker. And if you just, for example, move the MS2 loops around to the cytoplasmic marker, you only get the cytoplasmic protein made. Again, with a roughly 1 in 10, 1 in 20 suppression ratio. We want to do more. We want to go towards multicolor labeling. We want to catch up with what chemistry can do. So we thought on how when we put multiple organelles in it, let's not forget the key requirement whenever we put an organelle is that we don't screw up the host. No, we want the host machinery to still do amber codons as a stop codon, and only proteins that are made inside the organelle or at the surface of the organelle, only those should genetically encode a natural amino acid. That's the goal. So we thought of what's the best way to achieve the highest local concentration, because in the end, as I will show you, it boils all down to make a very sharp suppressor tRNA gradient in which 2 ribosomes can immerse. So we, genetically, we build autos in 2-dimensional ways, but also membrane anchoring them. And I show you the co-localization data for the plasma membranes. This is easiest to see. If you here do an RNA fish, a tRNA fish, you can see that we really build less than 70 nanometer rim of the suppressor tRNA. No, and the tRNA can diffuse, but it's bound to the synthetase, but the synthetase also lets it go. And the ribosomes can interact. We have also ribosomal markers here in one of the movies that I brought with me if you want to see it. And they can really go to the plasma membrane, and that is enough to generate an efficient system. So ultimately what we have now is a system where, for example, inside the hex cell, which is very small, you can build an auto on the plasma membrane, and here you can genetically encode amino acid X into protein Y with the AMBER codon. You have the cytoplasm where stop means stop, and that is what we want. We don't want to mess up the host. There's still enough ribosomes available in the cytoplasm. And we can, for example, have on the mitochondria another system that doesn't mix with this system, where we can suppress the AMBER codon to encode unnatural amino acid Y. And in a hex cell, mitochondria come with less than a micrometer of the plasma membrane system, so we were also surprised on how well this actually works. We thought this is a generalizable method. We've made, these blob-like organelles, the film-like organelles, where I just showed you the data, as well as these fiber-like organelles, where we engineered really things beautifully on the cytoskeleton. All of them actually work very well. So we thought, like, come on, this is totally generalizable. And then we did all of this for, the paralysing system, and we cloned the other common synthetase, and realized that, we get efficient genetic code expansion in all when you build an auto, but only our paralysing system is selective, mRNA selective. The others are not able to distinguish RNA with an MS2 loops from the other RNA. They're just as stupid as a normal cytoplasmic expression. That is, of course, not what we wanted. So we tried a lot of things. We, checked if the concentration range, the tRNA concentration range. Unnatural transition, we checked if, getting an NES on the synthetase might help that, because we've shown before that engineering the NES on the paralysing synthetase really helps it, because otherwise there's a lot stuck in the nucleus. We played with multiple copy numbers, and for a few of those tRNAs, we even were able to engineer the V arms of the tRNA to get, stronger binding and colocalization. And none of this works. It's even more data. Very, very frustrating. And very hard to publish negative data. I can only tell you what our working hypothesis here is, and that is that the K-cat for paralysing synthetase is orders of magnitude different to any of the other synthetases that are used in that field. And that allows for very slow turnover, and apparently seems to be a critical factor for generating a special tRNA gradient. So there's a 1,000-fold difference between the system that allows RNA selective expression and the other ones. And there's a little bit of happy ending thanks to Michael, who really did fantastic work on this, and that produced a lot of negative data, which I actually thought was, intellectually quite stimulating, but, still negative data is hard to publish. So he maintains the N-terminal domain, which gives rise to the binding and probably to the off rate, and was then able to swap through, a phenylalanine synthetase against the other domain from the paralysing synthetase. Also, that one gives us at least a second synthetase that works with autos, and into which, different unnatural amino acids can be genetically encoded. So, to come to an end, but before I do this, also come to the Oreo cookie. So we thought, what else can we do with that system? How well can we use this concept of, spatially engineering enzymatic function? And, their process, for example, pseudo-uridination is a process that allows to change, a base to pseudouridine, uridine to pseudouridine. And there are enzymes that can do this. For example, this DKC1 complex, it's a tetramer. And it's known to be able to, edit or modify U to pseudouridine. And if you throw this enzyme complex on the cell, it has some specificity already, because it has these aligning RNAs that these guide strands. But these tools people hoped at some point to use for gene therapy, because you can cure stop codon diseases with this, like mucoviscidosis, CFTR mutations. When premature stop codons show up and make you very sick, with those systems, people hope that they can cure people at some point, and then efficiency and selectivity really matter. Yeah. So what we essentially hoped is that we can make those systems even more efficient by basically, confining the enzymatic function to the plasma membrane and engineering it in a more specific way. So if we bring the key component to the plasma membrane, and similar as for translation, we don't need to recruit also ribosomes. We just need to recruit the essential factors, and the rest has accessible to membraneless system or an open system, and they're bound here to membrane. So the idea is that inside the organelle, we get pseudo-uridine modification at the target site, while in the cytoplasm, that doesn't happen that easily. Yeah? And to cut a long story short, also in the interest of time, we developed a fluorescent reporter to report for this, and we got the signal that we wanted. We also confirmed this with a variety of sequencing methods that building an orthogonally RNA editing organelle, so an Oreo, is actually a viable way to enhance the specificity of this DKC1 complex in pseudourination. And with that, this is basically just a summary of what I said. I want to thank my group, the people who did it, and you for your attention.
That was great. There was a lot in there. Thank you very much. I like that you're crushing Coke. Is that Coke? I have a question about your MS2 loops that you use, and you made it seem like, you put them in the 3' end of the mRNA and you got it to work. So obviously there's some spatial considerations that need to be taken here. And did you play around with putting those hairpins, I should say, in different positions of mRNA and get different effects by doing so?
Yeah, that seems to be a robust thing. So we didn't play with it too much. We also got the other typical RNA targeting system, the Lambda 22, to work on first attempt. The system seems to be very robust. I mean, I think it's important to put it in the untranslated region, because otherwise, if you don't put it in the untranslated regions, you would get a modified protein, and that is, of course, less attractive. This one is really a traceless method from a protein point of view. And we haven't studied it to death because we didn't see with the things that we played an impact. Yeah? So within the space that we explored there, we didn't see it. And we did a lot of modifications when we tried to fix the other synthetases. Yeah? That was really where we really tried to get things to work and played with more copies and V-arm binders, and that was not the problem.
Yeah, it's interesting. I'm intrigued by your pseudo-U modification and thinking about making it more effective by targeting to the membrane and
more specific. Not effective. I think that's a very big difference. I think the best we so far got is recover efficiency of 100%. We haven't got things more efficient. And that's also not to be expected to be. I think, the best you can expect is as if you do cytoplasmic expression, because then you make use of all molecules in the cell. But maintaining RNA selectivity, that is what worked also for the Oreo.
Thank you for clarifying. There's some questions online I guess I could ask them. So great talk. Have you looked at recording target proteins and encoding non-canonical amino acids to other sense codons?
Yeah, we're working on sense codons. Yes. There was also a science paper from another group not too long ago, which had a little bit in there. I don't think there's a reason why this should be limited to stop codons.
Yeah. Synthetic organ film upper limit specificity in terms of presence of translation inhibitor in normal and serious pathological conditions can be evaluated? That's the question. I don't know if you can read it. It's actually on the Q&A.
Yeah, I'm not 100% sure if I understand. I can also go on this one. We haven't done much disease investigation yet. No, for the OTOs, we certainly haven't touched this at all. For the Oreos, we have worked on, you know, classical amber suppression models of, you know, potential clinical relevance, because as I said, the most interesting application of that specific system is to take stop codon out and stop codon diseases. Yeah. So we did show this for 1 or 2 proteins that are commonly used, that you can edit the stop codon out that way. For the OTOs, I have to say that, of course, we're doing something else. We modify the membrane now, so this might that might impact cellular signaling. We haven't seen strong effects for many of the OTOs. Actually, the first OTO that we did, actually, whenever you touch the cytoskeleton, we actually see an effect on morphology. But whenever we went to the membranes, we didn't see a morphological effect. That doesn't mean that it can't have impact cellular signaling or something like this. And then, you know, this is now for us also to investigate, you know, what problems come from that method. But it does solve the amber suppression selectivity problem. And depending on what you want to do afterwards, this can be a concern or not a concern that just needs to be, you know, investigated.
Yeah, I know that you said that you're not getting into therapeutics too much, but you're obviously are tinkering around and thinking about it. There's quite a bit of moving pieces that are involved with the pseudo-U in the stop codon modification. Have you thought about, are you playing around, are you getting into delivery and how you can make that effective?
Yeah, I mean, we would, for our method, to actually work in a human, you would need to deliver a modifier to the DKC1, basically. But that's all you would need to do, no? And if you take, you know, RNA vaccines now, the template, you can bring in proteins. Yeah? So the modification that we do here, I mean, I guess you would need to bring in the RNA guide that also you have to do anyway when you want to do pseudouridine editing. And you would need to bring in something that allows DKC1 to condensate. Yeah. So you would need certainly an RNA much longer than the guide, so much longer than 20 base pairs, needs to be somewhat protein-like, but that is possible. No, I think the corona vaccines are 250 base pairs or so, and with that you can start thinking of ways on how to achieve that.
Yeah, that's great.
So one of the reviewers said, like, it will never ever have any application. And that I mean, of course, we're very far away from it, but I'm not sure I would subscribe to never ever. So I think the problem of the DKC1 system versus the problem of the DKC1 system plus an Oreo, that is not the biggest step. Yeah. And if the problem of the DKC1 system makes it into clinic, and as with many gene or RNA therapies, people do die of off targets. That's one of the biggest problems. If you can get an order of magnitude enhancement or suppression of off targets, and for all of that, you just need to put in a second RNA into your lipid protein complex to do this in human. I think that is actually, yeah, it's a technical issue that should be solvable with existing technologies. Yeah.
Yeah. No, I agree. Any other questions from the group? Doesn't look as if there are. You want to close this out, Rick?
Yeah, that sounds good. Well, we'd like to thank Dr. Lemke and May again for such wonderful talks. We really appreciate having you this month, and hopefully we'll see you again here soon. And thanks everybody for joining us.
Thank you, everybody.
All right. Bye-bye.